Our next presenter is Dr. Marci Drees. She's a board certified in internal medicine and infectious diseases and is the infection prevention officer and hospital epidemiologist for Christiana Care. She's a clinical investigator with the Center for Outcomes Research and co-director of resident research for the Department of Medicine. Dr. Drees serves as the chair of the Infection Prevention Committee and serves on the Healthcare Associated Infections Advisory Committee for the Delaware Division of Public Health. Marci, take it away.
Okay, thank you. You can hear me okay, and you can see my slides, correct?
I can hear you and I can see you.
Okay, so thank you to the organizers for inviting me. I'm actually going to turn off my video so it doesn't interfere with my Wi-Fi. And so I'm going to talk about COVID-19 immunity and vaccines today. All right, I do not have any conflicts of interest. I do want to say, of course, that the data that I'm presenting are accurate as of yesterday when I turned the slides in, and Dr. Henderson already alluded to the fact that there is new information out today. So I am going to talk about that, but it won't be included in the slides. So what I'm going to talk about first is give a little bit of an immunology primer. I needed it myself. I'm not an immunologist. I'm going to talk a little bit about natural immunity to COVID-19 infections. I'm going to talk about the process of developing a COVID-19 vaccine, as well as talking about a few of the early the vaccine candidates that are likely to come to market first. I'm going to talk about what the processes in place are for vaccine safety monitoring, and then just a little bit at the end about how do you go about implementing a COVID-19 vaccination program. So, Immunology 101. So I think many of you probably remember from your training that we have two different types of immunity known as innate and active immunity. Your innate immunity is that quick, non-specific immunity that acts really quickly to remove infectious agents, and your adaptive immunity is really more long-term and more specific to the individual pathogen. And there are a number of different cells involved with some overlap, but overall the innate immunity is those dendritic cells, macrophages, granulocytes, whereas your adaptive immunity really relies on your B cells and T cells. So again, your innate immunity, the idea is that when a pathogen enters the body in whichever method it does, those initial cells recognize that as a foreign body. They release cytokines and interferons. Those cytokines and interferons do a number of things. They signal the neighboring cells to defend themselves. If cells that are already infected are signaled to die, and then they recruit white blood cells to stimulate that long-lasting immunity. So, but as we all know, sometimes because this system is quick and rapid, it can overshoot. And so we hear about, the cytokine storm, and what we're seeing with COVID is sometimes it's that over-aggressive immune response that's really causing the severity of illness we see with COVID. So ideally, we want our adaptive immunity, right? So again, that antigen approaches the antigen-presenting cell, often a dendritic cell or a macrophage, which then stimulates those CD4 cells to differentiate. And they can differentiate into CD4 helper cells, of which there are two types, Th1, which stimulates the B cells to produce antibodies, and Th2 stimulates B cells. And Th1 stimulates stimulates macrophages and other cells to directly attack. And then they can also differentiate into killer T cells, or CD8, also known as cytotoxic T cells. So that first pathway is really what's called our humoral response, the antibody response, whereas the other pathways are more of the cell-mediated response. And we're going to see that both of those responses are important when it comes to COVID immunity. I also wanted to talk briefly about the fact that all antibodies are not the same. So there are neutralizing and there are non-neutralizing antibodies. So a non-neutralizing antibody, can bind the target on an infected cell, and that stimulates a natural killer cell or other cell to then recognize that infected cell, which then causes the cell to die and prevents production of more virus, right? So that's a good thing, but unfortunately the cell has to die to do that. The other pathway shown on the right side of this diagram, which is that when a B cell is able to release neutralizing antibodies, those neutralizing antibodies actually bind up the pathogen before they even infect the cell, right? So then the cells don't die, and it's more effective. And so when I present later in this talk some of the data for the specific vaccine candidates, I'm going to talk about both binding antibodies, which are the ones on the left, the non-neutralizing, as well as the neutralizing antibodies, because they're both really important. And then this is just a diagram of what we expect happens as the adaptive immune response to SARS-CoV-2. So you can see there's both a T cell and a B cell response. You can see it takes a little while, 1, 2, 3 weeks, depending on which type of memory cell, which type of antibody we're talking about. And the antibodies themselves, whether it's IgM and IgA or the IgG, they do peak and then they start to decline over time, and that's normal. Antibodies have a specific half-life. They don't remain in the body forever, assuming that you're not constantly being stimulated by that pathogen. But of course, what we all hope for is that we have that anamnestic response that's shown on the right side with the dotted lines, which of course we don't know yet because we haven't had experience with COVID for a year or longer yet. But we all hope that that's, once that immune system has been primed, whether it's from natural infection or from a vaccine, that you will have that response to prevent reinfection. So again, when I talk about the vaccine candidates later, these are the things you really want to pay attention to. So not only what is the overall antibody response, those binding antibodies, but what is the neutralizing antibody response? And then they're also looking for making sure that the vaccine is stimulating a CD8 T cell response, as well as a CD4 T cell response. And within the CD4s, whether it's a Th1 versus Th2 response. And the reason that's important is that if the Th2 response seems to be predominant, that actually can increase risk of what's known as vaccine-mediated enhanced disease, or VMED. And what that means is someone who's been vaccinated, if later on they're exposed to the pathogen, they actually have a worse outcome than if they were not vaccinated. And obviously, we do not want that to happen. So that has been something that all the manufacturers and the FDA and ACIP have been very attuned to. Okay, so what do we know about natural infection and immunity? So Dr. Henderson earlier talked about some of the other human coronaviruses that we see. And so I'm going to first talk about non-COVID-19 coronaviruses. And we know that, the normal human coronaviruses cause the common cold, and we really only see transient protection when people are infected. And so we think that over time, those antibody levels wane, and then you're susceptible to that same virus again. Although it's felt that if you are infected again with the same virus, your infection may be less severe or even asymptomatic, and typically associated with lower viral titers. What do we know about SARS-CoV-1? So this was a small study, but it followed patients with SARS for 36 months. And they found that the SARS-specific IgG and neutralizing antibodies peaked at month 4 and then started to diminish. But they could detect both those binding and neutralizing antibodies in 70% and 20%, respectively, in patients by 3 years after infection. So certainly it seems like those antibodies were persisting longer than for the other human coronaviruses. And then for Middle East Respiratory Syndrome, or MERS, again, a very small study, but they found that antibodies, including neutralizing antibodies, persisted in the majority of people at almost 3 years. So there may be an inverse relationship between severity of illness and persistence of antibodies. So what do we know about human SARS-CoV-2 infection? So I'm going to show you a couple of slides that show, so this one shows that, serum antibodies do decline between your acute phase and your 2 months post-discharge. And they looked at both asymptomatic and symptomatic patients. But again, this is very much expected because antibodies have a normal half-life in circulation. This study, again, very small, but they looked at 15 hospitalized patients, and they looked specifically for neutralizing antibodies. And they did 88 samples collected between day 0 and day 75 of when they developed symptoms. And they really found little to no decrease over 75 days since the symptom onset. And the blue line in this graph kind of shows the average that really showed that those antibodies really persisted at least for 75 days. But of course, like I said before, it's not only the humoral response, we also need to know what the cellular immune response is. And so this study looked at not only the antibody levels, but the memory B cells. And you could see that the antibodies on the left do decline, as I just showed, but those memory B cells really persisted over the duration of this study, which is exactly what they're supposed to do, right? They're supposed to be maintaining that memory of that pathogen so that if you're ever exposed in the future, you can ramp up much more quickly. And then this study looked at and they just found that people who have recovered from COVID-19 have both specific CD4 cells as well as CD8 T cells. So we do seem to be generating not only antibodies, but a cellular immune response from natural infection. Dr. Henderson talked briefly about reinfections, and I'm not going to really go into that in any sort of detail. This data was presented by the ACIP about a week and a half ago, and if you you can find those slides online if you are interested in delving more into this topic. But basically, there are very limited data, and you can see there's, the one at the top is the strongest evidence, and it goes down from there of patients that they feel were truly reinfected. So it does seem to be a very limited situation, at least early on after initial infection. So in summary, for the COVID-19 immune response, we believe that repeat exposures to SARS-CoV-2 may boost your immune system. Several studies have demonstrated waning of serum antibodies after a few months, and the implications for protection are really unknown at this time. However, neutralizing antibody titers have demonstrated little or no decrease at 75 days, and they have detected cellular B and T cell responses in COVID-19 patients, with memory B cells not waning as fast as antibody titers. And a reinfection with SARS-CoV-2 is possible, but does appear unlikely, at least within 3 months. To move on to what are the viral vaccine targets? So Dr. Henderson also showed you this picture as well. And I'm showing it again to emphasize that the target really for all of at least early candidates for vaccination is that spike protein, which contains the receptor binding domain. And this is another graph of what that looks like. The receptor binding domain is shown in green, and the entire spike protein is shown there on the left with a couple different configurations of it. And I believe all of the 4 vaccines that I'm going to talk about today all code for that entire spike antigen protein, not just the receptor binding domain. So, but it's important to know that really they've plucked that bit of genetic material out of the virus genome to put into these vaccines. So none of these vaccines contain coronavirus or any sort of whole genome of the coronavirus. So what is the vaccine development process? And I think there's been a lot of concern, among the public in general about how fast it's going and that things are being rushed. So I wanted to spend some time talking about that. So what this graph shows, and what it doesn't show is all the preclinical work, the animal models, everything else that has to happen before you can even take a vaccine candidate to the first human trial. But your phase 1 trial typically is in 10, 20, up to maybe 100 healthy volunteers, and it really is a dose-finding study, as well as they're looking for serious side effects and they're looking at antibody response. If that all looks good, sequentially they'll go to a phase 2 study, which just has 100 volunteers and also looking for the most common side effects. And really the only outcome that they're looking at is the immune response, those antibody levels. And these are the data I'm going to show you a little bit later. And then if that all looks good, you can move on to your phase 3 trial, which is typically thousands of volunteers. 30,000 to 45,000 is what is currently being studied. And that is very typical for a vaccine trial. These are not small studies, they're expensive. And again, you're looking at not only is the vaccine safe, you're going to have a better chance of detecting side effects when you study more people, but now you're not only looking at antibody responses, but you're looking for, does this vaccine actually prevent clinical disease? So that's the difference there. And then once all those trials have been considered, the FDA will license the vaccine only if it's found to be safe and effective, and the benefits outweigh the risks. So this is really what the overall pathway looks like. So again, that clinical development piece, that is what is happening in the pharmaceutical and the vaccine manufacturers. For COVID, there's been close coordination within Operation Warp Speed, or OWS, which I'm going to talk about in a second. Once they have finished that clinical work, they will submit to FDA for either a full licensure or an emergency use authorization. And it's likely at this point that it will go through the EUA process. There is also a third pathway called expanded access. It's unlikely that any COVID vaccine will go through that process because of all the additional paperwork required for the IND applications. Once the FDA approves a vaccine, then it goes to the ACIP, the Advisory Council on Immunization Practice. This is a CDC committee who reviews all that evidence again, and then they will make a decision as to whether to recommend the vaccine for use or not. And they do not have to match what the FDA has approved it for. So there really are two independent steps. And then once the ACIP recommends it, the CDC will then recommend it. But it really doesn't stop there. There is always post-approval monitoring, and those data are fed back to ACIP and FDA, because we know that as a vaccine goes out into the population, that's when you're going to detect the particularly rare side effects, and we need to make sure that we're keeping an eye on that. I want to talk a little bit about the ACIP itself. So it consists of 15 voting members that have various expertise that's relevant to vaccine administration and planning. It is very important to know that this is independent of CDC. The voting members are not CDC employees, they're not pharmaceutical employees, they are scientists, academicians, researchers who have expertise in vaccines and vaccine planning. And similarly, the FDA has its own advisory committee that is very similar. It's independent from pharmaceuticals as well as from the government. ACIP also has other representatives, 8 ex officio members that represent other federal agencies, and then 30 liaison representatives that are non-voting, who represent other societies with interest in vaccines. So I am the SHEA, Society for Healthcare Epi of America liaison, which I've been in that position since June. And it's kind of how I got all involved in this. So the ACIP meetings are all public. They've been virtual since June. They typically have only 3 meetings a year, but this year, of course, has been very different. So they've had daily, all-day meetings every month, basically since June. And it's important that not only are they open to the public, anyone can sign in and view the meeting as it's happening. They also must have time for public comment. In addition, for COVID-19, there's been a vaccine work group that has had weekly meetings really since April. What's presented in the work group is confidential, and they've really set the stage they review all the epidemiology, modeling, ethical principles, the specific vaccine candidate data, and then they come up with the framework to present to the overall ACIP for vote. And the framework that has been used, I think, since 2018 is what's known as the evidence to recommendation. So we review all the evidence, not only what's published, but what's known as the gray literature. And they use a very structured framework called the ETR that looks at is this a public health problem? What are the benefits and harms? What are the does the potential vaccine recipient value this vaccine? What is the acceptability, feasibility, resource use? And then what's been added for this one is equity. And that will, I think, be part of that framework moving forward. And then they can then provide to the ACIP the recommendation either to recommend for everybody or for a particular population without further discussion, or recommend for certain individuals based on shared clinical decision making, or not recommending. And it was decided very early on that each vaccine candidate will be reviewed on its own merits, not this, unlike say flu, like they talk about flu as a group of vaccines, even though there are some differences within them. Each of these vaccines is going to be reviewed individually. So it's been a little bit confusing keeping track of everyone that's been involved in the COVID-19 response. So there are 3 governmental entities that really have distinct roles in this program. So the first I already mentioned is Operation Warp Speed. So that's the US governmental body that's really responsible for the strategic approach, the coordination, and most importantly, or very importantly, allocating resources to make this happen. In addition, there's the Accelerating COVID-19 Therapeutic Interventions and Vaccines, known as ACTIVE, which is a public-private partnership with NIH, and they're looking not only at vaccines, but at therapeutics and everything else. And then the COVID-19 Prevention Network is the NIH-funded networks of, to help coordinate the phase 3 trial execution. So why is this important? So, we have a number of different candidate vaccines, all on different platforms. And if they are left to just, run independently, it will be very hard to compare one to the other. So the idea with COVID-19 Prevention Network is that they're all harmonized. So they are using similar methods, similar definitions, similar testing. They have a common data and safety monitoring board, or DSMB. And the whole idea is that once all the work is done, it will be easier to compare candidate vaccines to one another. So this is a slide from Operation Warp Speed. And first of all, so the departments that are really coordinating this together is the Department of Defense and the Department of Health and Human Services, which includes CDC, NIH, FDA, and BARDA. And the idea is that the typical process shown at the top there, it says 73 months to completion, but it can take years, 5, 10, even 15 years to bring a candidate vaccine from its initial preclinical work all the way to market. And obviously we don't have that luxury with COVID. So the idea here is that you can overlap some of these stages. So your preclinical and your phase 1 can overlap a little bit, your phase 2 and phase 3 can overlap. But the important part there is that brown line, which is the manufacturing process. So, again, I mentioned before, this is bringing a vaccine or really any new drug to market is an extremely expensive process for a company to do. So typically you would not move to your phase 2 trial until you were very sure that your phase 1 looked good. Likewise, you wouldn't move to phase 3 until your phase 2 analysis was complete. And then you wouldn't start manufacturing until all that clinical work is done. What they're doing here is they've started manufacturing really as soon as the phase 2, phase 3 trials are started. So Operation Warp Speed has given literally billions of dollars to some of these pharmaceutical companies. And honestly, and many of them have fronted that on their own as well, to really start manufacturing, even knowing that the science may not pan out. And that if the phase 3 trial does not show that it's a safe and effective vaccine, those doses are going to get thrown away and never distributed. So it's a huge financial risk to move things forward this quickly, but it's not a safety risk to the extent that I think people tend to think it is. So ultimately, what do we want a vaccine to do? So there are many steps in the process, right? So you would want, if you're vaccinated, would you want, you may want to be protected from even getting the infection. If you're not protected from infection, well, at least you don't develop any symptoms. If you develop symptoms, maybe you're not hospitalized or end up in the ICU, or hopefully, you don't die from the infection because you've been vaccinated compared to if you weren't vaccinated. And then of course, we all want something that can prevent transmission, that would help, really cut down on the spread through the community. So, but in terms of planning a vaccine trial, it's really difficult to detect asymptomatic infections, right? As well as, how do you really measure transmission in a community? So that's not really practical for a clinical trial. Likewise, if you're trying to prevent the more severe infections or the hospitalizations or the deaths, those luckily are relatively uncommon compared to the total number of infections that we see. So you would really need much, much larger trials to be able to power specifically for those outcomes. So where we've really landed with is the main outcome for all of these vaccine trials is the ability to prevent disease, defined as symptomatic COVID-19 that's PCR confirmed. Well, where do we really want to get? So of course, we want to achieve that herd immunity, also known as population immunity. And the idea there is that if you can reach a certain threshold of immunity through vaccination, you can protect the entire population. And so not every single person needs to be vaccinated. We know that not everyone wants to be vaccinated, but more importantly, not everyone can be vaccinated, either because of underlying medical conditions or other contraindications. So as long as we can achieve that threshold, we can protect everyone in the community. And so what is the threshold that we need? So it really varies based on the disease. We know with measles, which is probably the most contagious virus that we have out there, we need a very high level of vaccination, 95%, to achieve herd immunity. And we know, we've seen in the past several years that when there are pockets of communities that fall below 95%, that's when measles starts to spread. Polio is less contagious, so its threshold is about 80% vaccination. Where's COVID? Well, it's estimated between 50 and 70% of people need to be immune to order to achieve that herd immunity. The unfortunate part, though, is that it's estimated that currently, looking at the U.S. as a whole, our population immunity is less than 10%. There are probably pockets that have had high infection rates that have higher population immunity, but overall it's less than 10%. So really the only way that we can get to from less than 10% to 50 to 70% is through vaccination. You know, it would have untold cost in morbidity and mortality to let you know, the disease, even if we try to limit it to, you know, younger, healthier people who are unlikely to have severe complications, it would not be ethical to do that. And there's a quote by the WHO, which I felt sums it up quite nicely. So Operation Warp Speed has set, you know, phase 3 efficacy trial principles that all the manufacturers need to follow. So the sample size of approximately 30,000 volunteers is recommended, initially looking at those over 18 years of age. And they also are asking for an enriched subset of those who are at higher risk of disease. So if you only study the vaccine in, you know, 18 to 24-year-olds who don't have any underlying medical conditions, that really limits the generalizability of your results. So they are including people that are at higher risk of severe disease, diverse populations, and the elderly. The primary endpoint, as I mentioned, is the prevention of symptomatic COVID-19 that's virologically confirmed. And they really want the primary endpoint to be at least, or efficacy to be 50% or greater with a lower bounds of 30%. And that's consistent with what the FDA has said as well. And so again, they want everything to be harmonized. How are we measuring immunogenicity, and to make sure that these trials can be compared. And again, the common independent DSMB that's managed by NIAID. So when the COVID-19 work group began working, so they really had 4 guiding principles. One is that safety is paramount. Vaccine safety standards will not be compromised in order to get to a vaccine faster. They wanted to make sure that the clinical trials were inclusive, that we plan for efficient distribution, but yet we maintain flexibility. So while we want to have some national standards, they want state and local jurisdictions to have some flexibility when it comes to actually administering vaccine. And underlying all of that is equity. Because as Dr. Henderson mentioned, and I'm sure as you all know, COVID-19 has not affected all groups equally. And so we want to make sure that they have equal access to vaccination. All right, so now I'm going to get to the specific candidate vaccines. So right now there are more than 200 candidates in clinical development across the world. 47 are in development, 47 are in clinical development, and 155 are in preclinical development. Within the United States, there are 4 vaccines in active phase 3 and 5 in phase 1 or phase 2 clinical trials. And these are the, there's a number of different platforms or strategies that are being investigated. And the 4 vaccines that I'm going to talk about today, from an interest of time, are the mRNA and the replication defective viral vectors. So why are there so many different platforms, right? So unfortunately, there is no one perfect answer, right? So the DNA, RNA vaccines, have the advantage of being very fast, and which is why we're seeing those 2 RNA platforms really first, likely to be first to market. Other ones have other potentials, maybe a possibility for a single dose. They may be more scalable than others, or they may be platforms that we've actually used before, so there's a little bit more confidence in potential for side effects and that sort of thing. So there are pros and cons to every platform. And so overall, though, I think it's good that we have so many different options that are being investigated. And certainly the hope is that we'll have not one, but many different vaccines eventually be successful and come to market. So these, as of the end of last month, these are the COVID-19s that are in human clinical trials in the US. The 4 that I'm going to talk about are the 4 that are currently in phase 3 trials, but for completeness, you can see the other ones. And likewise, there's a few others that are still in early trials in the US. So the first 2 I'm going to talk about are these 2 RNA vaccine candidates. And so because this is a new platform that really hasn't been shown before or used before, I just want to spend a minute talking about how it works. And so that's shown here on the right side of the slide, where again, they've taken that the RNA from that spike protein and they've put it into a lipid coat, which is then injected. The lipid coat allows it to enter cells, where that mRNA is used by the normal cell mechanisms to create viral proteins that are, again, for that spike protein specifically, that are then expressed and generate that immune response. So they're pretty safe, they're pretty easy to develop, which is why they've come along more quickly. But again, we just don't have any experience with them. So the first one I'm going to talk about is the mRNA 1273, which is the Moderna vaccine. So again, it's mRNA, no prior experience, and it codes for the full-length spike protein. Logistically, it's a little challenging. It has 2 doses, 28 days apart. They settled on a 100 microgram dose as the optimal dose. It will be shipped in multi-dose vials with 5 doses per vial. And once you've entered that vial, you need to use the rest within 6 hours. There is no preservative or reconstitution required. Storage does have some storage issues long term. It needs to be stored at -20 degrees Celsius. But once you've thawed it, it can stay at refrigeration temperature for about 2 weeks. So that is that one. The immunogenicity, this comes from their phase 1 trial, which again was a small trial of 45 adults, although they did enhance it to include some older adults as well. And I think it looked pretty promising. So that pink bar across the 2 graphs, the binding antibodies are shown at the top, the neutralizing antibodies at the bottom. The pink bar shows kind of where we would expect someone to be if they were convalescing from COVID-19 vaccination, or sorry, COVID-19 illness. And you can see that really across all age groups, at 36 days after vaccination, people developed a sufficient antibody response that was comparable to natural infection. So that's very promising that it worked as well in older as well as younger patients. And they did also notice a Th1 biased T cell response. Again, so to help, that would predict a less likelihood of that vaccine-mediated enhanced disease. Well, what about safety? So this graph shows a lot of the systemic as well as local side effects. First for the first dose and then the second dose. The gray are mild, the blue are moderate reactions, and the grade 3, the orange is the grade 3 or severe. So you can see, particularly after, you can see it varies by different age as well, but you can see especially after the second vaccination, there is a not insignificant percentage of people that are complaining of symptoms. However, are primarily mild, and there were no severe adverse reactions seen, or no hospitalizations seen after these in these trials. And I think you'll see that this is a common theme for all of these vaccines, that they are fairly reactogenic, meaning they do cause symptoms after vaccination, but the vast majority of them are considered mild. So Moderna actually published some of their clinical trial inclusivity. So I don't have this type of data for the other vaccines, but I just wanted to give you as an example how they are really striving to include a diverse population. So you can see the racial and ethnic makeup there on the left. More than a quarter have comorbidities, including diabetes, cardiac disease, lung disease, and obesity. About 22% are healthcare personnel. And you can see the age distribution there on the right as well. So they are attempting to really enroll a diverse population, and the other companies as well are doing the same. They just have not published their data yet. Moving on to the Pfizer BioNTech disease, BNT162b2. So another mRNA vaccine, again, codes for that full-length spike protein. This one is also 2 doses, but is 21 days apart instead of 28 days like the Moderna. The dose that they settled on was the 30 microgram dose. Also will be in multi-dose vials with 10 doses per vial. And I'm sure you all have heard that this one is the one that's generating a lot of angst around its storage. So the problem with mRNA, which is probably why we haven't seen it being used for vaccines in the past, is that it's not a very stable molecule. So it really needs that long-term or that cold storage to even keep the vaccine viable, the mRNA viable. So that's part of the problem that we're seeing with these particular vaccines. But this vaccine needs to be stored at -70. They do come in a storage container with dry ice that you can keep for up to 10 days in that storage container, as long as you're able to replenish the dry ice. Once you've taken it out of cold storage, it can stay at refrigeration temperature for about 5 days. It does have to be reconstituted in order before administration, and then you have to use it within 6 hours. So again, a lot of logistic challenges with this vaccine, which is looking now like it will probably be the first vaccine that comes to market. As far as its immunogenicity, I apologize, this is a complicated slide, but that's what's publicly available. But this is from the phase 1 trial, and they were looking at 2 different doses. I've cut the slides so we're only showing the candidate vaccine that actually went to the phase 3 trial. And then the red boxes show the dose that they actually took to the phase 3 trial. So you're comparing those levels. Again, the top is the binding antibodies, the bottom is the neutralizing antibodies, and you're comparing it to that gray bar on the right side, which is your convalescent plasma. So you can see again at 28 to 35 days after vaccination, this vaccine is generating a very high antibody response for both binding and neutralizing antibodies that's similar to human convalescent plasma. And they also, I'm not showing this data, but it also demonstrated a strong CD4 and CD8 T cell response with the Th1 dominance. Now, again, as far as safety, the top row of graphs are for the 18 to 55-year-old age group. The bottom are for 65 to 85-year-old age group. And again, you can see really across the board, whether you're looking at pain or more systemic symptoms like fever, fatigue, chills, a significant proportion of patients, more so in the younger age group than in the older age group, are complaining of symptoms, but the majority are mild. All right, moving on to the next 2 vaccines. These are both known as viral vector vaccines. And so the idea here is that you're using that virus, which in both cases is an adenovirus, really as a Trojan horse. So you've taken the virus, you've taken out the viral genetic material, and you've put the coronavirus, spike protein genetic material into that package. So the virus attacks or infects the cell as it normally would, and then it expresses those spike protein antigens that then are secreted in various ways to generate an immune response. The first one I'm going to talk about is the AZD1222, which is the Oxford University AstraZeneca consortium. This one uses an existing simian or chimpanzee recombinant adenovirus vector known as ChAdOx1. It is a non-replicating virus, so the virus will not continue to produce itself once it's injected into the body. And it really, because humans are not exposed to this chimpanzee adenovirus, it doesn't normally infect humans, that really circumvents any pre-existing immunity that we may all have to various adenoviruses. And there is prior experience with this ChAdOx1 vector. They've developed influenza, chikungunya, MERS, malaria, TB, Zika, and meningitis B vaccines with it. So there is a reasonable experience, at least as far as safety goes, in using this type of vector. And again, it contains the genetic material of the spike protein. This one is also a 2-dose vaccine, 28 days apart. It contains 5 × 10⁻⁻⁻ viral particles, which, as you can imagine, is a lot of virus because it's not replicating itself. So you have to give people a lot of it to generate that immune response. Also in multi-dose vials, 10 per vial, and once you've entered that vial, you need to use it within 4 hours. However, this one does not have that cold chain requirement. It is stored at refrigeration temperature, 2 to 8 degrees, and I think it can stay at that for an extended period of time. As far as immunogenicity, this again is from their phase 1/2 randomized controlled trial. They actually compared it to a meningitis vaccine instead of placebo. But you can see again that on the far right in the green is the convalescent plasma. And you can see that a single dose resulted in the spike protein specific antibody by day 28. That's the prime graph in the middle. And then 10% participants did receive a second dose, which is shown there in the prime boost. And they saw a further increase in that spike protein specific antibody, which is why they decided to go with a 2-dose regimen. And they also describe a robust Th1 biased T cell response. And this is their neutralizing antibody data. The 1-dose regimen is shown on the left, and the boosted regimen is shown on the right, which again shows a further increase in antibody levels after getting that second dose. As far as safety goes, this is they showed it giving it with paracetamol or acetaminophen at the top and without at the bottom, a fair amount of pain and tenderness, but again, mostly mild. And then this is the more systemic symptoms, fatigue, headache, malaise, muscle ache were all reported fairly commonly, even if you gave paracetamol. But again, most of those reactions were mild. And last but not least, I'm going to talk about the Janssen or J&J vaccine. This uses a different adenovirus platform known as AdVac. It is adenovirus 26, which does not commonly circulate in humans. And the whole idea there is, if you use an adenovirus that people have already been exposed to, you may not generate the same level of immune response to the vaccine. So you want to choose something different that people hopefully don't have experience with. This is also a non-replicating virus. There is extensive experience. More than 110,000 patients have gotten an AdVac-based vaccine with the different diseases listed there. And specifically, pregnant women and children have received this vaccine in large numbers with no significant safety concerns. So that I think is important. And this one, again, also includes the full-length membrane-bound spike protein. The major advantage that I see to this one is that it's 1 dose. It also contains a large number of viral particles. It will also come as a multi-dose vial that once you've entered, you have to use within 6 hours. This one does, for long-term storage, is at -20 degrees. But once you've thawed it, you can actually keep it at refrigeration temperature for up to 3 months, which makes the logistics a little bit easier. This is also their phase 1/2A trial, where they looked at both younger adults, 18 to 55, as well as those 65 and older. They looked at 1 versus 2 doses in about 1,000 participants. And so again, on the far right in gray is that human convalescent plasma comparison. And so you can see they really generated very similar both binding shown at the top as well as neutralizing antibody shown at the bottom after 28 days post-vaccination. They also showed a robust CD4 and CD8 T cell response that was Th1 biased. So in that case, they really didn't see a big advantage to the second dose, which is why they're going with the 1-dose regimen. This is their safety. And again, this is all blinded. So you can see all participants in the younger adults at the left and the older adults on the right. Still, a very significant percentage of patients with local and/or systemic symptoms, but the majority of which were mild. So what is the timeline? So that's the $1 million question. So the Moderna had announced late last month that their enrollment of 30,000 participants was complete, and the majority of them had received their second vaccination. Pfizer had also announced at the end of last month that they were at 42,000 participants enrolled out of a goal of 44,000. So they were very close. And again, the majority of them have received both doses. You may have heard on the news that both AstraZeneca and Janssen had to be on a FDA hold/safety pause, respectively, for a while. They were both lifted at the end of October, and so they are resuming their phase trials. And it is important to recognize that that is a very normal part of the process, and I don't think does not bode ill for either of those two vaccine candidates. And I think it does show that the system is working as far as monitoring for safety. So they have pre-designated interim efficacy time points. So when they're looking for that, again, that clinical efficacy of at least 50%, it was determined that Pfizer would have to look once they had seen 32 cases of COVID. And again, they don't know whether those cases were in the placebo or the vaccinated arm, and 53 cases in Moderna. As Dr. Henderson showed earlier, so they waited until they had seen double that. I think they had seen 64 cases of COVID before they went to the FDA. And then by then they'd actually already seen another 30. So they ended up doing their interim analysis based on 94, I think, cases to show that 90% efficacy. And so Moderna is still waiting, presumably, to see their 53 cases. It's a smaller study, so they need more cases to show the applicable power to be able to look. And basically what happens when they reach that number of cases is that then their DSMB can unblind the data. The pharmaceutical companies themselves cannot, to look to see what the breakdown was between vaccinated and placebo, to see if it's worth going for EUA at that time or just continuing on the study. All right, vaccine safety monitoring. So I'm going to go over this pretty briefly because I don't remember how much time I have left now since we started late. But there is, as far as the clinical trial monitoring goes, the FDA has released very specific guidance for those phase 3 trials, which is that they need 2 months median of follow-up in order to go for EUA. So that means that at least half of the vaccine recipients must have had at least 2 months of follow-up after completing both doses. And is that enough? I think, on the one hand, adverse events that are plausibly linked to vaccination generally start within 6 weeks after receipt of a vaccine. However, the FDA typically requires 6 months of follow-up for a full licensure for any new vaccine. And there's the question is, some of these are including new vaccine technologies, and should we even be following them longer? The FDA, in this article that was just released last week, just feel like the shortened time frame is justified based on the fact that we have extensive historical experience with adverse events after vaccination, which again typically start within 6 weeks. Obviously, we don't have the luxury of time to monitor longer, in that thousands of people are dying every day from this illness, and so there is some urgency there. And given that the magnitude of vaccine effectiveness that's required to support a favorable risk-benefit analysis profile under an EUA is pretty high, right? So an EUA for a drug that you're only giving to the sickest of the sick patients is very different from a vaccine that you're going to be giving to millions of healthy people. So this criteria are actually pretty high even under EUA to move forward. The US government actually has a long-standing program for vaccine safety monitoring, and the US government is responsible for public safety related to vaccines. So the monitoring is independent from the manufacturers. It covers all vaccines and is in addition to the fact that each manufacturer is required to have their own safety monitoring plan. And the government maintains a large, robust, and sophisticated safety monitoring system that requires collaboration among multiple government agencies. And in addition, the ACIP has established the COVID-19 vaccine safety technical subgroup. And so I'm going to very briefly talk about a few systems that we're using for passive surveillance versus active surveillance of vaccine safety. So hopefully you all are familiar with VAERS, the Vaccine Adverse Event Reporting System. It's the nation's frontline system for monitoring safety. Its strengths include the fact that it can really rapidly detect safety signals and can detect rare adverse events. However, its limitation is because there is no control group, it really cannot assess causality, but it can really, again, show that safety signal that can then be investigated using other means. And there really is a critical role for healthcare providers here to report any potential vaccine adverse event. It does not have to be proven that it is related to the vaccine. It can be anything, can be reported. So you all are aware of that. And there's a lot of information on how to report on the website. I'm going to skip over this, but just so you know that there is this clinical immunization safety assessment project, which will actually help support U.S. providers around vaccination issues. The links are there, and you'll receive the slides after the talk. There is also the FDA has what's known as the BEST system, which they partner with multiple partners that are listed there that represent a variety of healthcare settings, and they include electronic health record data, claims data, and some systems that are linked between claims and electronic health, overall covering more than 250 million individuals. And again, they will be using that system to assess and confirm potential adverse events or safety concerns for COVID-19 vaccines. The FDA also has a longstanding partnership with CMS to look at the older population that are Medicare beneficiaries, more than about 55 million people. And it's very representative because the vast majority of those who are over 65 use Medicare, so it's not a random sample, and it covers a variety of healthcare settings. It includes both claims data with access to medical charts, and they'll be doing what they call near real-time surveillance or rapid cycle analyses. And they've been doing this with flu and Guillain-Barré since 2007, and they'll be monitoring, between 10 and 20 different safety outcomes of interest for COVID-19 vaccines. And those specific outcomes are yet to be determined. There is also the Vaccine Safety Data Link, which also is 9 participating integrated healthcare organizations covering more than 12 million people annually, and they will also be doing that rapid cycle analysis, looking for, among other things, the evidence of vaccine-mediated enhanced disease, and they will also be looking at issues about safety during pregnancy, including fetal death and outcomes. And then last but not least is a new system known as vSafe, which is a new smartphone-based active surveillance program for vaccine safety. It will use text messaging to initiate web-based survey monitoring. So as someone is vaccinated, they can be enrolled in the system, and they will be given information about enrolling. They do have the opportunity to opt out, but they will be getting texts every day for the first week after vaccination, and then for 6 weeks, with additional health checks up to 12 months after their vaccination. And should they develop any clinically important events, that they will be directed to report to VAERS. So it's really, VAERS is a very passive system, but this will help make it more active. All right, last but not least, a few slides on vaccinating or implementing a vaccination program. So ACIP has adopted some guiding principles around, what should you be thinking about as you plan your vaccination program. Obviously, maximizing benefits and minimizing harms, promoting justice, mitigating health inequity, and promoting transparency. And again, the slides from this presentation are available on the ACIP website. But within each of these categories, there are specific questions that you should be asking yourself as you're planning your vaccination program under each of these topics. So transparency, I think, there's so much concern about how this has been rushed. I think, that can probably not be overstated, that you just need to be very, very open in what you know and what you don't know as we move through this process. And so, of course, we are expecting a phased vaccination approach where initially we will have, very limited doses available. And so, very constrained supply, which will require highly targeted administration. And, the example populations that are there, phase 1A is considered likely to be healthcare providers as well as first responders. Phase 1B may be other populations. And I think that's where it gets a little bit trickier. Do you hit high risk conditions, older adults, other essential workers, that sort of thing. In the middle, there will be a large number of doses available with sufficient supply to meet demand. And so then you really have to work on broadening your access. And then eventually this will likely be more routine. As we plan, the one thing we know is that there are many unknowns, right? So we don't know for sure what vaccine is first, although we think it will probably be the Pfizer. And there's just many unknowns about, we don't know how long the immunity will last, whether this will become an annual booster type thing like flu. There's a lot of unknowns. But again, we anticipate that one of these mRNA vaccines will be first. If it is the Pfizer, there are significant logistical constraints around the cold chain, the multi-dose vials, the fact that you have to reconstitute the vaccine, who's going to do that, and where. So you really will need to have high volume throughput in order to avoid wasting vaccine, as well as the fact that 2 doses are needed. So you have to have a method to notify people when they need to come back for their second dose. So much more complex than our annual campaigns. Approval under the FDA EUA is likely. And what will happen is once the manufacturer submits for that EUA, the FDA will hold an emergency meeting to review the data. Once the FDA approves the vaccine, then the ACIP will hold an emergency meeting to decide whether to recommend it and to whom and for whom. We do not have any recommendation to make this vaccine mandatory. It's one thing to, when we have decades of experience with flu vaccine or MMR is one thing versus a vaccine we have only a few months experience with. We do recommend that, because the vaccine is not mandatory, you don't have to cover any adverse events under workers' comp. So we do certainly recommend that you voluntarily cover this under workers' comp so that our workers are actually confident in that they'll be taken care of should they have any adverse events. And I already said healthcare personnel will likely be phase 1A. And it's important also to recognize that even once you're vaccinated, there will not be a change in our need for PPE or physical distancing. All those other tools need to really continue. Healthcare personnel is a very broad definition. It's paid and unpaid persons working in healthcare settings who are either exposed to patients or infectious materials. And so it includes not only direct patient care, but those who could still be exposed at work. And of course, some of them also have high-risk conditions. Some of them are also 65 years old or greater who are at higher risk. So how do you prioritize within that? So I want to point you to the link at the bottom, which is the Shea document that has a lot of these thoughts written out in more detail than I have time to go through today. So I encourage you to look at that document because it will help you think through some of these issues. But in terms of prioritization, those healthcare personnel who interact directly with patients or their family members are certainly higher priority than those who are able to work remotely. In my opinion, it really doesn't matter whether they are caring for COVID positive patients or COVID patients who are not suspected of having COVID. I feel like there probably is equal risk there, just because we don't have universal testing, and even if we did, it's not 100% accurate. And also, to really not forget the other healthcare personnel that are providing essential services throughout the delivery system, the environmental services, the escort, everyone else that's going all over the hospital. Again, ensuring equity in all stages of planning. Our lower wage workers may have higher rates of COVID for a number of reasons. They can't work remotely. They may need to take public transportation. They may live in different environments that expose them more in the community. And then, again, based on supply and demand, if the supply remains insufficient for the groups above, consider prioritizing those who otherwise are at higher risk for severe disease. As far as vaccine hesitancy, this has been an ongoing issue, and the CDC has a nice presentation that's referenced there at the bottom about some of the things to think about. I mean, what I'm considering is that, we know that for vaccines in general, the most deciding factor for patients is that a healthcare provider recommended it. And so from my perspective, I can't tell any of you to get a vaccine that I myself wouldn't be comfortable getting. And likewise, I can't expect you to tell any of your patients to get it if you're not comfortable getting it. So really focusing on making our healthcare providers comfortable that this is a safe and effective vaccine is really priority number one. Most people are somewhere in the middle, they're not flat out refusing and they're not flat out demanding it, but they really want a wait and see approach and want more information, which I think is perfectly appropriate. And so the CDC is developing this Vaccinate with Confidence national strategy. So it's more than just a media campaign, but it will be focusing on reinforcing trust in general, empowering healthcare providers, and engaging communities and individuals. And there will be lots of tools and other things forthcoming. Some of it has already started with the vaccine website, and that keep an eye on this website because they will be adding more and more in the coming days and weeks. All right, so that was a whirlwind review, and hopefully I'm not too much over time. But I would like to thank Shay for allowing me to be the liaison to ACIP. It's been an amazing experience. The members of the COVID work group and all the CDC and FDA staff and the many others who really are devoting countless hours to this response. And I think, to date, it's been a largely thankless job. So thank you again for inviting me.
Great, thank you, Marci, and really appreciate the overview of vaccines. There are a lot of questions and concerns regarding safety and efficacy. Many questions are coming in, and we would like to hold those questions for the panel because there will be an opportunity for the panel to talk about vaccine efficacy as well as safety.